This video will demonstrate how to align and compare the similarity of your fish sample CO1 gene to the human sequence. We'll compare both DNA and amino acid sequences, starting with DNA. You'll need to download the human cytochrome C oxidase 1 barcode region DNA and protein sequences from the Sequences page on Canvas. And you'll also have to have your sushi sample edited DNA and translated amino acid sequences handy as well. From the BLAST page, again select nucleotide BLAST as the search type to run. Again, copy and paste your edited fish CO1 nucleotide sequence into the sequence window. This time, check the box next to Align two or more sequences under the sequence window. A second subject sequence window will appear below. Copy and paste the human CO1 barcode region sequence into this second box. Under the heading Program Selection, choose Somewhat Similar Sequences BLAST N. And then click BLAST to align your sequences. When your result comes up, click on the project name. I didn't provide one here, so it says None Provided, or the Alignment tab to view the alignment of the two sequences. The top line is your query sequence, your fish sequence that you entered first. The subject line is the human sequence that you entered second. You can see the level of similarity in terms of identities, that's nucleotides that are identical between the two sequences here. As before, you'll want to capture an image of this alignment for your lab notebook. To align the fish and human protein sequences, go back to the starting page and select Protein BLAST as the search type to run. You're going to repeat the same steps, but using the translated fish sequence and the human CO1 barcode protein sequence as your query and subject. Again, choose Align two or more sequences, and for your second sequence, use the human. Leave the algorithm as it is and hit BLAST. Click the Alignments tab to view the alignment, and again, note the similarity in amino acid sequence and the percent identity. Make sure to capture an image for your lab notebook.